just to put this in a little bit of perspective, I started my career working on model reduction, things that had nothing to do with chemical reaction networks, using very old-fashioned kinds of techniques, invariant manifold methods, some singular perturbation theory. And we've kind of kept that line of research pottering along over the years. But Oh, it's almost 20 years ago now. Maya Minsheva, who's in this room today, was joined my group as a postdoc, and she got me interested in chemical reaction networks. And a couple of years ago, these two things just happened to come together. So let me tell you a little bit about the sorts of things I'm interested in and why they might be interesting and how they're connected to today's topic. So there's a lot of oscillators or clocks in biology. The circadian oscillator, which I'm sure most of us are familiar with in one way or another, especially if you travel. There's another very famous oscillator, famous because kind of its mechanism has been dissected in great detail, called the semitogenesis oscillator. And what it does is it helps form segments. Essentially, if you want to think of it this way, it's kind of an advanced version of that is what forms vertebrae in invertebrates, for example. Of course, we also have other kinds of interesting behaviors in biology. So we have an awful lot of multistability. In fact, cells of multi-cellular organisms develop, many, many, many cell fates from a single genome. So essentially, that's multistability, right? The biologists don't call it that. They call it totipotency or pluripotency or any of a number of other names similar to those. We also have lots of wave phenomena in biology. So in cell polarization, for example, there's a wave that spreads across the cell, but then there's an event called wave pinning that stops the wave spreading, and that creates an anterior and a posterior pole in a cell. And of course, kind of a classic thing, people have been talking about Turing instabilities as explanations for developmental patterning for a long time. Animal coat patterns is kind of the classic example, but we've also been looking at left-right asymmetry of internal organs, and I'll talk about that a little bit later. But there's some issues that we have to deal with in cell biology that are a little bit different than the ones we deal with in conventional chemistry. The models are large. Now, that's true of some chemical models as well. I've had some contacts over the years with the combustion community, and they have very large models sometimes. But there's no question that the models that we need to develop for a lot of cell processes are large, many, many variables, many complex interconnections between different subsystems. But of course, we have these large models, but we also often don't know the parameters, or we know very few of them. Very, very, very few of them are available from the literature typically. And those parameters that are available are often not transferable. And what I mean by that is if you measure the Michaelis constants from one type of cell in one organism, they'll be different in other organisms, and they'll be different possibly even in other types of cells in the same organism. And so, just because the Michaelis constants from a liver cell, you go to a skin cell and you're going to have different kinetics. And the models themselves, so often when we start a model in cell biology, we kind of know the rough way things are connected. We have some experimental data that tells us that one thing activates another or inhibits it. But we often don't have a lot of the mechanistic details really nailed down. So the models themselves are often subject to really significant uncertainty. In other words, we know kind of the large scale structure, but we don't know all the details of the reactions. And so the question that arises then is whether we can simplify a large model to create a smaller model that preserves the dynamics of the original without having detailed knowledge of the parameters. So I referred to these earlier methods that I had worked on. You always need to know parameters to do a calculation using these other methods. You can do a certain amount of stuff analytically, but in the end, whether or not that particular reduction is valid requires knowledge of the parameters. So what would that fix? Well, if we don't really know all the mechanistic details, then maybe it's okay if we blur some of them by having a simpler models. a simpler model. Of course, you end up with a model with fewer parameters, which maybe you can fit the data more effectively than a really large model with a lot of detail tucked away inside it. And so it would help a lot if we could do that. So shifting gears a little bit, of course, this group is very interested in reaction network theory, and that, of course, is connected to qualitative stability analysis in a bunch of different places. And there's a long history of that. And this is a very partial list, right? I could have started with Ruth and Hurwitz in the 19th century. And of course, there's been lots of work since the 90s that's been done, in fact, some of it by folks in this room. But there's a lot of methods, there's a lot of ideas. As I kind of hint below, I even pruned some branches here from this large tree of qualitative stability analysis methods, because there really is a really impressive literature in this area by now. And some of it's also outside of chemical reaction networks. I've emphasized things that have to do with reaction networks here, but there's a lot of theory in ecology and lots of other areas. Now, if you think about the structure of the qualitative stability theorems we get from these various branches of qualitative stability analysis, they generally take one of the following two forms. So either they say property P is necessary, and if you're lucky, sufficient, but often all it says it's necessary for behavior B. So an example of that from Ivanova's method, which is the thing that Maya introduced me to many years ago, is that you need a critical fragment, and I'll explain in a few minutes what that is, but a particular kind of sub-network that has this property of criticality of order n equal to the rank of the stoichiometric matrix if you're going to have bistability. And if you don't have that, you'll never have a bistable reaction network. The other way it's sometimes phrased is the opposite. Property Q forbids behavior B. So not Q is necessary for behavior B would be another way to put that. So an example, the deficiency 1 theorem, which I'm sure a lot of folks in this room know more intimately than I do. If a chemical reaction network and its linkage classes all have deficiencies of 0 or 1, the sum of the linkage class deficiencies equals the network's deficiency, and each linkage class contains a single strongly linked terminal linkage class, then the system cannot be bistable. So if you have these properties, no bistability, right? And so, of course, a bistable system would have to violate at least one of the conditions of the theorem, right? So again, not one of these properties necessary for bistability. So here's the basic idea. So what we want to do when we're going to reduce models is we're going to want to preserve conditions for the behavior that the model exhibits that's interesting. So typically what will have happened is, so one of two things. Either I have a claim from the biological literature that a certain set of connections gives me a behavior. So again, multistability, oscillations, whatever it is. And sometimes it's somebody will have worked up a model based on those hypotheses, but the model is very, very large and subject to all the uncertainties I was talking about before. So I either go in thinking maybe this will be, example, bistable, or I walk in knowing that it is because somebody's done some numerical simulations and found a bistable regime, but I still have all the problems of an excessively complicated model to deal with. So if I've got one of those situations, and I know that some particular property is necessary for the behavior, again, say bistability, then maybe what I can do is simplify the model while making sure I preserve the property P. So first I might verify that it has that property, and then I would make sure as I simplify it that I preserve that property. So similarly, if not Q allows for behavior B, then make sure you preserve not Q during model simplification. So I'm going to, because that's where we've done the most work so far, I'm going to talk a little bit about Ivanova theory. This will be very, very high level because I don't want to get lost in the details. But roughly speaking, in Ivanova theory, you start with a bipartite graph representation of a chemical network. You have two node types, reactions and chemical species. That's the bipartite part. You connect them with arrows that go from the reactants to the reactions, and then another arrow from the reaction to the products. If you have some stoichiometric, well, you always have stoichiometric coefficients, but kind of normally you only write down the ones that aren't equal to 1 because you just assume all the others are 1. And so stoichiometric coefficients are associated with edges. So in other words, the arrows that connect the thing. There's a way of constructing subsets of these graphs known as fragments, and the nice thing about fragments is each one of them corresponds to a term in the characteristic polynomial. There's a really simple one-to-one correspondence between them. And once you've identified the fragments, you can compute the coefficients of the coefficient, of the corresponding characteristic polynomial term from the structure of the fragment. So identifying a fragment essentially tells you what you should write in the characteristic polynomial. Now, a fragment with a negative coefficient is called critical. So we basically go and we look for specific things that we know tend to generate criticality, and we try to identify these critical fragments. And the reason we're interested in those is because critical fragments are necessary for bistability, and they're permissive of oscillations. It's the story with oscillations is more complicated. And if you're really interested in that story, you should ask Maya to tell you about it. All right, so that's the basic idea. So we're going to extract these particular pieces of the bipartite graph that are called fragments. We look for critical ones. The critical ones are the things that we really want to preserve. So the fragments themselves are made up of subgraphs, and if you want to calculate the coefficient of a fragment, you add up the coefficients of the subgraphs. So there's kind of a nested doll kind of feature to this theory that takes a bit of getting used to. But now if you dig into how you calculate the coefficients of a subgraph, it turns out that they really just depend on a couple of things. So one of them is how many cycles are there in the subgraph, and what are their parities? Are they even or odd? And the other thing is, the other thing that's important, so most of the stoichiometric coefficients in most networks are equal to 1, but if there are any that are not equal to 1, you need to be a little careful because they enter the calculation in a very particular way, and they can have a pretty significant effect on the result. Not too surprising, right? Because we know about things like autocatalysis where, coefficients of 2 are often quite important. So if we can leave those things alone, in other words, don't mess with the cycles and don't lose any stoichiometric coefficients, then we should be able to simplify a model and be okay. So I'm going to go into an example, and this is an example we worked out a few years ago. So Roussel and Roussel, the other Roussel here is Catherine, who's both my research associate and wife. And it's a really interesting system. So on the outside, we're quite symmetric, but on the inside, you're probably aware that your heart leans to one side and your two lungs aren't quite the same size and your liver is not centered and all those sorts of things. And that's quite reproducible, right? The heart in the overwhelming majority of people leans very far to the left. The left lung is consequently smaller than the right lung. So a lot of this stuff is entirely reproducible. And in fact, people who have inverted symmetry often have birth defects to go along with them. So how does this internal asymmetry arise? Well, we know it has to do with a protein called nodal, which I've got in, I'm not sure, can you see my hand, my little hand that I'm waving over my graph here? Yeah,
Yes.
okay, excellent.
Yeah.
So this protein called nodal binds to a cell surface receptor during embryonic development. What that receptor does is it phosphorylates a protein called SMAD2. SMAD2 forms dimers, and those dimers assemble on a scaffold called SMAD4, and that's the transcription factor that moves into the nucleus and causes transcription of a couple of genes in particular. So one of them is nodal itself, so there's an autocatalytic loop there. And the other one is another protein called lefty, which is an antagonist. And as the name suggests, as an antagonist, it essentially blocks the cell surface receptor that nodal normally binds to. And it's this kind of delicate dance between lefty and nodal that's thought to generate left-right asymmetry. Now, the interesting thing is that lefty is mostly expressed on the left side. So originally people thought lefty was the determinant of of the left side fate. But it turns out it's nodal, and lefty is, in fact, an antagonist. And that's because both lefty and nodal, again, are transcribed from the same, the transcription factor, kind of activates the same, both of those genes, I suppose I should say. So this is not by cell biological standards a particularly large model, but it was big enough to be awkward in some ways. 18 chemical species. There's 5 conservation relations. So the order of the model is 13, which is kind of medium sized, I guess I'd describe it. But we were interested in kind of understanding kind of some of the properties of this model and to what extent do they depend on details. So really quickly, this model is bistable, and that allows for a bunch of different properties, including wave propagation. We're still looking at that. So what you're seeing here is a stable steady state. This low branch here would be a low nodal right-hand side kind of state, and then this high branch over here is a high nodal left-hand side state. We're varying one of the particular rate constants, but of course, if you've done these things before, you can vary almost any of the rate constants and see somewhat similar bifurcation diagrams. Right. So when we went looking for critical fragments, we tend to adopt a strategy where we try to look for the smallest ones first, even though technically for bistability, it's kind of at the higher end you're looking for things. But it turns out that if you identify a small critical fragment, you can kind of bootstrap it into a bigger critical fragment fairly easily by adding edges. Anyway, that's a technical detail. So the critical fragment is exactly what you might guess. It's the autocatalytic loop. So here's nodal. Nodal activates the cell surface receptor. This piece over here is Michaelis-Menten catalysis of the phosphorylation of SMAD2, and then SMAD2 dimerizes. Not shown here because it's not part of the actual fragment. Phosphosmad2 assembles on SMAD4. You get your transcription factor. It activates the nodal gene, and the nodal gene makes more nodal. So it's the classic autocatalytic kind of loop. Now, when we looked at this, and this was before we'd really thought about using these methods for model reduction, it was just an observation as we were going along. If we remove these highlighted vertices here in red and just reconnect from reaction 9 to the gene, which is, of course, kind of non-physical, but it helps us kind of understand, again, what's important in this model. The fragment is still critical. Why? Because we haven't removed any cycles. We've just kind of removed a few pieces from a cycle. They all have stoichiometric coefficients of 1, so they're not a problem. So I'd have to be careful about this edge over here. But removing this stuff doesn't cause any particular problem. So I haven't changed the number of cycles. I haven't changed the directions they go or anything else. And so I should be able to do that. And so here's what it looks like after you've done that. You just reconnect from R9. I usually change the symbol for the reaction once it's no longer an actual physical reaction, and reconnect over here. And the simplified model can be verified to still be bistable, which is kind of nice. And what this allowed us to do was to say, in the end, it's this dimerization of SMAD2 that's really important, and not, in fact, we looked at a few different variations. It's not kind of the order in which things assemble on SMAD4 or any of these other details that matter all that much. So that was great. So again, kind of model reduction, but model reduction in the aid of kind of understanding network dynamics, which was also kind of pleasing. And if you look hard, you can probably find other things you can imagine simplifying out of here. So I'm not going to do that, but just to give you, just to let you know that, you can do this thing iteratively and just keep removing stuff until you have a truly minimal model. So after that initial observation, I got a very good graduate student, Talman Soares, and we did a little bit of thinking about, well, can we generalize this? Can we actually write down a set of rules that allow us to simplify these models? And the short answer is yes. In fact, we developed a set of what we call conservative rules that can be carried out without even knowing what the critical fragment is. You just have this big network, and you start looking for things that you can simplify out that would be harmless. But if you happen to know the critical fragment, you can do some more aggressive reductions. And so, kind of you can imagine a strategy where you start with a big network where you don't even know the critical fragment because it's too complicated to go looking for it. You start pruning it away, simplifying it in ways that are known to be safe. And when it's small enough, then you can start to think, okay, where's my critical fragment now? So it opens up some possibilities to do some of these sorts of things. And the keys are things like don't change the parities of the cycles. Again, if it's an even cycle, don't make it odd and vice versa. Don't destroy cycles, which is surprisingly easy to do if you're not careful. And it's also surprisingly easy to create new cycles if you're not careful how you take things out and reconnect them. Um, and of course, be real careful with those non-unit stoichiometric coefficients. We have some work in progress right now with a new graduate student, Nadia da Silva, and we're kind of now going to look at how this generalizes using other qualitative stability methods. We're looking at the Thomas, the really instability theorems, and looking at kind of what we can do with those. They're very simple. They're based on just Jacobian cycles. And that makes them a good place to kind of test out a few ideas. So the questions are, can we eliminate unnecessary cycles, shorten cycles, and so on? And our preliminary results do show that you can do some of these things again. But again, you identify what's critical now in the sense of important to the behavior, and you make sure you hold on to those as you're doing your reductions. We do plan to extend, so Nadi is currently working on a proposal to extend this to other qualitative stability analysis methods, probably Feinberg style chemical reaction network theory. Um, uh, there's some interesting questions buried under here. So one of the things we found is that like formal rules to simplify the reaction graph sometimes lead you to things that are hard to interpret chemically. And so one of the questions that I have that I want Natty to think about a little bit is, can we come up with stricter rules so that the reduced models always make chemical sense? And the other thing I've been thinking is kind of once you've kind of reduced something to the smallest you can reduce it, does this give you a classification of oscillators and multi-stable models? Like a whole bunch of things collapse onto the same reduced models? There's been a bit of work on that sort of thing coming from the other end, kind of saying what are the simplest, kind of what are the smallest models with certain behaviors, but we want to start from the big end and say, does it boil down to a few simple cases? So I mentioned all of these folks before. Catherine, Talman, and Nadi have been all wonderful partners in this work. My work is funded by NSERC, and Talman had some funding from the university, which was very much appreciated as well. Thank you.